I want to welcome here Mark Davis from the INHS. Part of our spiel is that we get people to introduce themselves. And you say a little bit about yourself in your research. We're labeling you with eDNA, but I know you do a lot of conservation work. I know you've been one of the leaders here at INHS. So can you tell us a little bit about yourself, your lab, and your research, please?
Yeah, absolutely. Thanks, Matt, and thanks you all for joining. Thanks to you all for joining me this morning and for the invite. I really appreciate it. As Matt mentioned, I'm a conservation biologist here at the Illinois Natural History Survey, and I am the director of our collaborative conservation genomics lab, where we kind of do all things genetics and genomics, from more conventional phylogenomics to cutting edge conservation genomics, but we've also been pushing the envelope on environmental DNA, and that's been a large part of our research portfolio over the past decade. And so that work is cut across all taxonomic groups, all ecological contexts. We're working around the globe on these things, and it's a mix. And so environmental DNA, for those that are unfamiliar, really is only about 15 years old as a sort of conservation genetics subdiscipline. The first paper was published out of Europe about 15 years ago, tracing the invasive American bullfrog in wetlands in France. And since then, there has been a exponential increase of environmental DNA studies published, including the beginning of a journal in 2019 dedicated exclusively to environmental DNA, the Journal Environmental DNA. And so the work that we do is aimed in a lot of different contexts to do a lot of different things. And so it may be early detection of invasive species. It could be tracking and biomonitoring for threatened and endangered species. We've been doing work on whole communities. And it's a split. Some of that research is very much focused on the ecology of biodiversity. But we're also at the same time working on advancing the field of how to eDNA, because again, it is a relatively young subdiscipline. And so delighted here to talk to you all about all things eDNA this morning.
Excellent, wonderful overview. So you touched on the fact that you do things, we are the Illinois Natural History Survey. So we're focused, of course, on the Illinois, and we get a lot of funding, and our focus is there. But you touched on the fact that you're global in scope. So how
Yes.
important is it, and why is it critical to be global in scope when we're doing environmental DNA sampling? What about global is important?
Yeah, one of the things as a conservation biologist I drive my students nuts with is this notion that biodiversity does not respect geopolitical boundaries.
Yeah.
And so, restricting our field of view very narrowly to states, provinces, countries, oftentimes we are missing that perspective that, these species have a larger footprint. And with eDNA, this becomes particularly more pronounced. There's research out there that has shown airborne eDNA is detecting DNA from things that are moving through the jet stream and are being deposited from Southeast Asia here in North America. And so we have transport, and that kind of transcends all aspects. It just becomes, how does it scale? Right. And so you can have transport locally, you can have transport through watersheds, you can have transport through the wind, you can have transport through the airstream, through ocean currents, etc. And so oftentimes we have to think at that scale because that is the scale at which eDNA is moving at.
So how do you window those scales, Mark? Like imagine you're setting up
Poorly.
yeah, poorly. Yeah, I was just thinking like, okay, you want to do some compute and you want to be able to like say, okay, I'm going to start with a little window and then I'm going to get a bit bigger, bigger, bigger. I'm going to look for signal across those windows and so yeah, poorly. Like are there specific axes or parameters that you would that window create use for those windows?
Yeah, I'll say it's certainly context specific.
Yeah.
We often think in terms of watershed, so it's easier to think about in flowing water systems. And the problem that we run into is that it quickly becomes a highly multidimensional problem
Mhm.
because on the one hand, you have differential biotic and abiotic factors. So if we just
Mhm.
think about the biotic factors, different species we think have different shedding rates. Different species have different phenologies. And so you may be interested in, say, a mud puppy that is very active in the winter and the salamander mussel, which is more active in spring. And those two things are codependent on one another, and we're trying to track them both simultaneously because of their endangerment status. And so beyond the abiotic factors, there's a number of biotic factors. One of the things that's been keeping me up at night thinking about is this issue of DNA adsorption to substrates.
Uh-huh.
DNA is sticky, and we're starting to realize that the composition of your sediments can have differential impacts on how well it binds that DNA
Mm-hmm.
and can or cannot carry it along. Biofilms are binding eDNA on the substrate of our rivers and streams. You've got other factors like microbial action that is chewing apart that DNA that is influencing how far it's able to travel from point A to point B. UV radiation is really effective
Mm-hmm.
at degrading DNA. And so, how is UV exposure influencing that retention? And then there's just the interactions. We've been thinking a lot in our lab about pollinator eDNA, and we're finding increasingly that you can sit and watch a flower, and you can observe a bumblebee go into that flower and crawl around. And you can snip that flower off and try and harness the DNA from it, and you don't always or ever detect it. It seems that there's repeated visits that will build up that DNA. And so we're seeing this sort of bell-shaped curve where your more abundant species are better represented than those less abundant simply because you don't have that buildup. And so all of these factors conspire to our ability to understand fate and transport because it quickly, as you can see, becomes highly multidimensional
Mm-hmm.
and difficult to develop controlled studies to address all of them because we have known unknowns as well,
Yeah.
and nearly intractable in natural systems to begin to unpack. And so I don't have a good answer. We're getting better, and it seems that there's a mix, that we certainly have local signatures. I was really excited this week. A grad student, Olivia Reeves, that's been working in my lab, mentored by Eric Larson, presented her thesis research on Monday. And Olivia looked at forest patches in riparian zones here in Illinois, but didn't do what you think we might do. So Olivia wasn't particularly interested in what are the fish and the macroinvertebrates in the stream in forest patches in Illinois' riparian zones, but instead looked at what is the terrestrial community in those forests. And so taking water samples from the stream, thinking of streams and rivers as conveyor belts of upland eDNA. And what Olivia found is that we see a shift in those communities. So as you move from our grassland habitats, we see grassland specialists or more common species. And then the more forest cover, the larger the forest patch, the more we see those woodland specialists. So we are detecting things like flying squirrels, like bobcat, in the water that is being transported into the watershed. But again, in the interstitial spaces between these forest patches, we're not detecting those things. And so it suggests that there's a pretty strong local imprint. But then there's also the broader global pool that's being transported in. So it's super challenging, and it's difficult to say without, diving into the context specific aspects, what that transport and fate factor looks like and being able to really pinpoint.
Mm-hmm.
We're getting better, but again, important to calibrate. We're only 15 years into the eDNA revolution.
Yeah, it's really fascinating. I want to chew on this concept of, like thinking from a taxonomic standpoint, we've got kind of, when you go and you describe wasps from a morphological standpoint or a genomic standpoint, there's kind of these set things, characteristics that we have that we're referencing from the historical context. We'll discover some new things, etc. But concepts like these things that might be kind of more universal, right? Like everything has DNA.
Right.
We know that when we build global databases, I was familiar with some like the sex database, right?
Mm-hmm.
And they wanted to do it across life. Well, heck, they could only figure out like 6 or 7 things that they could actually measure because of, things are clones versus, sharing genes and stuff. There's very, very few things that we can measure for all species. We'll talk about species concepts next session. I'm really excited about that too. But like shedding might be one of those, right? Like all life has DNA. So is there some kind of thought about like, let's do a phylogeny and let's look for shedding as, the mechanisms or the modes of shedding across a phylogeny? What are the factors that, do mammals just, shed more than insects? And is anybody looking at that? How do things shed?
Yeah, that's a great question. It's something that I've been thinking about a lot, based on during the break, Deborah and I had a little bit of a conversation. She asked about soil eDNA, and we've been publishing a lot this year on using soil to track the federally listed eastern indigo snake in the southeastern U.S. And then soil, it's a tricky critter to track down, and there has been considerable literature that stems from, in my opinion, a rather dubious paper that was published several years ago, that reptiles don't shed DNA into the environment. So there's been a history of difficulties detecting reptiles written broadly, but turtles in particular. When we think about comparing amphibians to reptiles, we have this heavy keratinized skin. Those reptile species do shed that skin, but it's locked up in keratin. Very difficult if you've ever tried to extract DNA from a rattlesnake shed, like I have thousands of times. It's difficult to unlock that DNA. But I think what we're finding is that maybe not is not the case. It's that they do shed into the environment. It may be at lower rates. The problem has been our ability to develop the techniques, the methodologies to harness it. So it does seem like there are differential shedding rates. How that cuts across the phylogeny has not been explored to date, because again, it's going to require, I think, probably mesocosm experiments on a species by species basis.
Mhm.
You've got to have an idea of the biomass. And when we're deploying this technology and these methodologies in the wild, so to speak, we don't have that luxury necessarily of knowing just how much biomass is out in the landscape. Now, I'll throw in the chat later. We did publish a great paper here a few years back with Ethan Kessler in the PACE lab. Ethan had this luxury where he was reintroducing alligator snapping turtles into southern Illinois, and every animal in the system had a radio tag on it. We had biomass amounts, and what we found is that eDNA copy number in environmental samples tracked really well with upstream biomass. And so the more upstream biomass we had, the more eDNA we were detecting, to a point. As soon as that stream left our protected areas, the signal attenuated due, we think, to UV exposure. It just
Uh-huh.
broke apart that
Uh-huh.
signal very quickly. And so unless you either have a mesocosm experiment where you have a known biomass, or a system like Ethan's, which was brilliant in that we had every animal radio tag, we knew how much mass was out in
Yeah.
the riverscape, it's very difficult to really pin down what those shedding rates are.
Mhm.
And it's exacerbated in a number of contexts where, even some of our better studied systems, we still have gaps in our understanding of the biodiversity there. And this becomes compounded in places like the global south, where when it comes to our metabarcoding, we're
Mhm.
getting OTUs that we can't assign to species. And so
Mhm.
that creates challenges as well, where we have that shortfall in many cases of that fine-tuned understanding of the biodiversity and contemporary survey data to go along with it.
Yeah. Feels like there's an opportunity for sort of the more the traditional taxonomic folks who are going to go and create a concept that you could link to, right? Build out some metadata on that. So you're out in the field and you're like, you do some late night diagnostics and you're like, uh-oh, we've got like a lot of diversity. We just hit a little gold mine. We got a couple nuggets here. Let's bring in the team that will kind of quickly do some of that analysis and give you some anchors that you can take. Maybe we don't have a name for this thing yet, but we know we're in a little hot spot, and it feels like there's some synergies that could come out of that kind of work.
Yeah, I think there's an excellent opportunity for this positive feedback loop where, we can be out sampling in places and generating metabarcoding data where, we are going to detect some of those known, probably more common species, but we're also
Hmm.
going to
Hmm.
identify some OTUs that are not assigned.
Yeah.
And based on kind of that curve of how many species we can assign versus how many are unassigned, can give us some ideas on, all right, these are places where we can target conventional sampling
Hmm.
to bring in that
Yeah.
taxonomic expertise to identify those species. And I think more critically, and I hope that the biggest point I want to make today, and I empower you all to think about, is what we most need is associating with those species that we're identifying mitogenomes. That is a critically important piece to this puzzle. We tend to target mitochondrial DNA for our eDNA studies. Why? There's simply more mitochondria in a given cell than there is nuclei, right?
Mm-hmm.
And so we believe that there's more mtDNA floating around in the environment for us to access. But we also know that some of the primers we're using, number one, for metabarcoding have biases. And so being able to move beyond simply barcode data to other regions of the mitogenome are going to give us flexibility to have much greater resolution. Number two, it's going to build up those repositories. So my ability to be good at eDNA, particularly in
Mm-hmm.
metabarcoding, is only as good as the sequence repositories that are available to me. And so we have that luxury here. I mentioned Olivia's work did really well in identifying Illinois species pool because we have a good backbone infrastructure in place. But my preliminary work in Vietnam, in the Mekong Delta, very different. There is a massive amount of undescribed biodiversity there, and so our ability to, number one, identify those things to the highest taxonomic levels is attenuated, and number two, for some of the things about conservation, it also impacts our ability to actively conserve this biodiversity in a meaningful way. And so, finally, when we move away from metabarcoding, we're often thinking in terms of targeted qPCR assays to look for very specific components of biodiversity. Well, we have to develop those assays, and we need those sequences available to be able to do it effectively. And so by generating mitogenomes, it allows us to target different regions of that mitogenome to develop the most sensitive, accurate, cost-effective assays. And so again, I think there's an opportunity here to, maybe use metabarcoding to identify those hotspots of undescribed biodiversity,
Mm-hmm.
conventionally sample, bring in specimens to museum collections, and then in the spirit of the extended specimen network,
Mm-hmm.
have those mitogenomes associated with those described species as well. And that, for
Yeah.
me, is like the most exciting frontier on the back end of eDNA, is being able to build out those databases.
Yeah. So let's pivot to that a little bit. That's what we think about a lot is building out the informatics tools, and TaxonWorks sort of has up to we have a sequence model in our system, but there's no UI for that, right? So we're aware of how that kind of fits into that vouchering, the
Mhm.
extracts. We're going to have a really nice provenance chain that go from specimen to extract, extract to extract. Even if it's on, we'll be able to put in the anatomical parts in there. So say a little bit about, maybe to start off with, how do you manage your data, Mark? And particularly, where are the pain points with respect to, like, I wish I had X or Y? And maybe touch on, too, lots of different topics, how your lab members, we talked earlier on in the week about people learning from their mentors and then contributing data to a knowledge base that grows over time, over your career. How do you teach your students to capture their data, move it into your systems? How does that persist over time? Just say a little bit about the informatics of the Mark Davis research world.
Yeah, that's I would say, as someone that's constantly evaluating how we're managing our data, that I manage it poorly, because the challenge that we have is that uh, I'll be a little bit crass. My PhD advisor referred to me as a bit of a taxonomic whore. I don't have like a specific lane that I fit in. And
Uh-huh.
so, right now we have eDNA projects on freshwater turtles, on terrestrial snakes, on pollinator communities, on all of these different things in different taxonomic groups and different ecological contexts, that the methods that we're developing the data are the same,
Hmm.
but the sort of buckets they fit in are a little bit different.
Hmm.
And so, the management certainly is on a project by project basis, or maybe a kind of question by question basis.
Hmm.
So, I am working to develop our repository for pollinators, for example. We're doing a lot of work at pollinator eDNA in managed lands across the globe at this point. And I think that is feeding into a more global perspective, as now I've got projects stood up using the same pipelines, looking at the same sorts of species, using the same primers
Hmm.
to begin to develop a sort of regional, I guess, continental scale database.
Hmm.
Others tend to be one-off. We've got a project on rare turtles in Minnesota, and so those are sequestered away. And then we have the differential between the types of data. We've got metabarcoding data, which are from the Illumina sequencer. So sequence reads are the raw data that we're working with. But then we have qPCR data, which may just be ones and zeros, or it could be copy number. And so those two pieces don't really talk to one another necessarily, even
Mm-hmm.
though sometimes we're using the same samples.
Mm-hmm.
But I think that's where the connective tissue comes in, is that we're really tracking sample by sample. And so our negative 80, after they've gone through the process, our DNA extracts are stored in there, and every sample has an ID tag that is then associated with the various forms of data that we have generated with
Mm-hmm.
that specific sample. And so from a global perspective, I'm trying to track it on a sample by sample basis, because the beauty of it is that we can repurpose those samples to a point down the road. And so being able to track, number one, what's been done, what primers we've used for metabarcoding on those samples, what maybe qPCR targeted assays we have run on those samples, and then as we move forward in the future, we can then add on to those as we see fit.
Mm-hmm. Yeah, thanks for that overview. Um, all good
There's a bunch
stuff.
of questions there. I don't think I answered
Yeah,
all of
yeah.
them, though.
No, so I see. Yeah, no, I think it's okay. I'm getting we're getting a bunch of questions in the chat, and there was one that I think we could tie into that. You talked about putting things in the 80-degree freezer, right? So
Yeah.
we're preserving things, and you mentioned that there's a lot of like degradation. You're working on this time scale where the samples that are out in the field, you have to start preserving them, and it's got their half-life is really ticking, right? Like they're degrading, and your samples all have considerations like, okay, we've got to get this into the fridge. We only got a certain amount of time to get this DNA. It's just like this time scale that, people who go out and collect a bug and it's just not going to degrade, right? Like they're doing a
Right.
morphology, that old school. But you've got all these time constraints and all these processes that you need to record the metadata on. So Yuta asked a question along that line. One question regarding eDNA from Asia transported by jet stream to the USA, Illinois. How long would such molecules be in the air traveling? They should be strongly exposed to UV, correct? I'm wondering why you can detect them while eDNA a couple of kilometers downstream, the signal of forest patches or turtles is petering out. So just, talk a little bit about that chaos of the half-life of what's going on.
Yeah, that is a great question. And I think that for me is the really interesting aspect of it. And it gets really down to how we do eDNA, right? And so if I say I got a jar of pickled rattlesnakes in my lab and I want to get a mitogenome out of them, or maybe just a barcode sequence, yeah, we can preserve those in ethanol. DNA is really stable. I can take that, do a QIAGEN extraction, and in a couple of days, I'm getting the sequences back. Um, you can kind of take that same concept to eDNA in that it is going to originate sometimes as cellular DNA, sometimes as extracellular DNA. And so what we have are really nice long fragments of DNA in the environment that then degrades over time. And so for these wind samples, it is entirely probable that they're starting off as nice long strands. By the time they're reaching their final destination, we're talking 50 to 200 base pairs in length, right? So going from kilobases down to, I don't know, decibases, that's probably the wrong word I just made up, but very short strands. And so we're not using, when I'm targeting, say, a rare turtle in Minnesota, I'm not using the Fulmer primers to target several hundred base pairs. We're designing assays that are looking at maybe at most 200 bases.
Uh-huh.
Ideally, we're talking 125 bases. So we're looking at much smaller fragments, which presents challenges. And again, not to beat this dead horse, but speaks to the need for mitogenomes, because we need to be able to find places for certainly our targeted qPCR assays that within a taxonomic group have conserved flanking regions, but among sympatric species, they're variable, and so we need to have that information. But that's why you can still detect DNA at very long distances, because there is a non-zero number of DNA molecules that are not completely obliterated into our single nucleotides. We've got small fragments that we can harness.
Yeah. So let's flip that. Has anybody looked at knowing that DNA maybe break down according to some rates, right? So do they go and sample the eDNA and then do some sort of, CSI, like, when did the thing die or how far away? Like maybe there's a time. There's also like the factor of how fast it breaks down, but maybe we could predict distance or, predict something about the spatial nature or the chronology of the events based on the condition of the DNA.
Yeah, absolutely. And so I'm really excited. Back in the fall, Ellie Snyder joined us, postdoc on an NSF postdoc fellowship. Came home again. Ellie did her bachelor's here at U of I, worked in Ed DeWalt's lab at INHS, worked in my lab for a summer as well on eDNA. And Ellie's been doing a ton of work on measuring fate and transport. And so the fun thing that we can do, this is the fun stuff we do as eDNAologists in the summertime, you can go out to a stream and you can spike in synthetic DNA. So we can buy gene fragments from IDT, we can spike that into a stream, we
Very
can take
cool.
these
Yeah.
variables, and we can look at transport. And if you really want to get crazy, you can do some of the addition of dyes to look at how morphology of the stream might impact these things. You can do this in wind tunnels. You can do this in a lot of different scenarios.
Ah,
So
very
yes,
cool.
we're actively engaged in that, and so we're getting a better handle on how eDNA moves through and degrades, particularly in stream systems. Um, and Ellie's been really leading that on her work at Notre Dame, but contemporarily, and one of the most exciting new horizons in environmental nucleic acids is environmental RNA. And so that's what her work is doing here at INHS now, and others are doing this around the globe, is looking at the sort of fate and transport of eRNA. Number of reasons why that's going to be really important. If we want to go that direction, we can talk about it. But certainly that work is being done. And so this is why I mentioned that we've kind of got this split in the eDNA universe right now, where a lot of work is being done to inform or learn about ecology of biodiversity, distributions, things along those lines. But we're also working on the real nuts and bolts of how eDNA flows through our environment,
Mhm.
what it means for understanding the distance relationship between a detection and where that organism actually occurs, but also how all of these various factors, this multidimensionality that I spoke about, is influencing our ability to be rather informed on the kind of how and where and how what that half-life of eDNA is.
Yeah, very interesting, fascinating. So Debbie asked a question, and I'm going to twist it a little bit too. But do all these sequences end up in GenBank, and does this link to the sample and eventually the vouchered specimens? Do the eDNA community students learn the important and value the voucher and how to voucher? So I guess I'd add to that question is, how much do you put a trust in a mitogenome, for example, that's not like something that you know where it came from, right? Is there this like I can find all these mitogenomes, therefore I trust them, or
Sure.
do you need to see that voucher? And yeah,
I mean,
thoughts
for
along those lines.
me, as someone whose dissertation work was kind of reconstructing the phylogeny of the Western rattlesnake complex, I'm well aware that you cannot trust what's on GenBank. I think we all in this room know that there are sequences that are misidentified, that are misassigned, that can be problematic. And so that I think is also the truth for these mitogenomes. And so for my students, we teach the trust but verify.
Mhm.
These have had to go through a process to be accessioned into GenBank, but you've got to visualize your data. And if they don't make sense, then you might want to question that. And so, yeah, we absolutely emphasize the need for that verification, that validation. But again, this points to a problem with eDNA is that, again, in the long count of history of genetics, we're very young.
Yeah.
And industry best practices are still being established. And so it was only recently that a former postdoc in my lab and a bunch of other collaborators published a paper on best practices for the design and validation of our targeted qPCR assays that included a lot of work on how you verify those sequences by doing a lot of in silico testing. And
Mhm.
so the establishment of these rigorous workflows is coming online, but we're kind of just checking things off piecemeal. And as one mole pops up and we whack it, another mole pops up, we got to whack that one.
Yeah.
So it is a bit of a moving target. And so I think I'd flip it on y'all, because what actually we do is we rely on our museum repositories either for the associated sequence data that have already been generated with those vouchered specimens, or leveraging those tissue banks. And I'll use an example that we have here on our Minnesota Turtle Project. One of the issues that we ran into is that there were published assays for blandings and wood turtles. We
Okay.
deployed those in Minnesota at places where we had known telemetered animals and got no detections. Why is that? The reason is that those original assays were designed off of a very narrow regional sampling of turtles on the northeast extent of their range. And as we all know, you have genetic variation as you move from north to south, east to west. And so the assays that were designed did not capture that genetic diversity. We had mismatches in those primer regions that were leading to these false negatives. And so we were able to leverage tissue samples here from INHS and from other museum collections to then generate those data, accession those sequences now into GenBank to make them available for others, but also design really sensitive, accurate assays from it. And so for us, when we can go to a collection that has a vouchered specimen and tissues that we can access along with it, that vastly expands the opportunities for us to do the work that we do and to be
Hmm.
really good at it.
Yeah. Yeah. So I'll get back to another question, but I want to get Debbie's had her hand up for a long time, and maybe there's another question from her as well. Debbie, you want to just take over a bit?
There we go. Thank you for that, Mark. So I guess I'd at a later time pick up the conversation about what's the connection between collection of eDNA and the voucher? In
Hmm.
other words, if you need things to be vouchered, are the things being vouchered, or do your students also do that? Or do you have them reach out to the collections to say, hey, we need these things to be vouchered? Are they there? So there's some conversation there about who's doing what and what you need in order to do what you just said. That would be interesting to me. The question I
Yep.
wanted to ask to follow up on the eDNA was about viral data. So the looking for the host associations and pathogen preparedness kind of conversations. Is that also part of this eDNA work?
Uh, I do want to go back to your other question first,
Got it.
and I will say, as an old school skin out herpetologist, no, with eDNA we are not vouchering specimens and working with taxonomists. We are going out and sight unseen sampling to collect water samples, soil samples, air samples to document biodiversity. So we are not actively engaged in sampling for biodiversity. I think where we can come into play, and we've touched on earlier, is that I think we can inform where our blind spots are. So where are taxonomic blind spots that are precluding this methodology, this tool from doing as good of a job as it could. And so I think that's really where the rubber meets the road because I'll take, for example, I guess one example where we are doing this, right? So, wrapping up a project up in Alaska, we've been very interested in cloudberries. They are a
Mm,
culturally significant plant in the Yukon-Kuskokwim Delta. There is a great deal of concern that climate change is going to either negatively impact cloudberries and or their pollinators, but very little is known about their pollinators.
mm,
And so our team spent a couple weeks up in the YKD last year eDNA sampling cloudberry flowers to document those arthropod communities that are occurring on those. But also because we know we have some taxonomic blind spots, we conventionally sampled. My collaborator in Alaska
mm,
is describing those species, keying out those species. We're generating barcodes here because no one gave us money to do full genomes, and it's a little bit
mm,
more expensive than barcodes.
mm,
We're doing the barcoding here; those will all be accessioned. And so sometimes we are doing that for sure, but it's not the norm. I think that one of the things we like about eDNA is that we don't need those collection permits to go out and put up a dust trap and collect air samples. And so it's reducing those
mm,
administrative hurdles that so many of us spend so much of our time with.
mhm, mm,
But the second question was, what about viruses? And I would encourage you to go to the Entomological Society meetings this fall, where I've been invited to talk about some really exciting work that we're doing in tracking pollinators and their pathogens on flowers using both eDNA and eRNA. So yes, we are doing that. Absolutely. We've got plates running, I believe, right now, as a matter of fact, generating data. And so, yes, we can do those sorts of things. We've got a paper in the works now where we tracked both the Louisiana pine snake, the rarest snake in North America, as well as snake fungal disease using the same eDNA samples. And
mm,
so really context specific. It depends on the types of studies people are interested in, depending on what our funders want to fund. But one of the, I think, most common things that I say about eDNA, eRNA is that the only limitations are your imagination. If you think it's important to track a species and its pathogens, in theory it can be done, and in practice we're increasingly showing that it is effective.
Thank you. We'll talk more.
You bet.
So we got a question from Anna. Anna, you mentioned pollinator communities. How likely is it that eDNA could be used for monitoring pollinator populations over time, measuring both richness and abundance, perhaps, if possible?
Yeah, it's absolutely feasible. We're doing it right now. And that's one of the exciting things about eDNA to me is that we're deploying this in context where, for example, a parcel of land has been purchased through mitigation funds and is going to be restored. And so we can go out, we can sample that before the restoration efforts take place. We can generate that baseline biodiversity, probably multiple visits. Again, we want to be mindful of species phenology and environmental drivers of activity patterns, but you can do some comprehensive baselining before a restoration effort takes place. You can then go in during the restoration and then sample for a period of time afterwards to look at changes in those communities in time to assess does the promise of restoration match up with the data? Do we see those returns on investment? How long before we see those returns on investment? So yeah, absolutely. I'm really excited about a project that we're popping off this summer where we're working in solar installations and we are monitoring pollinator communities there where they're using different management practices to try and promote pollinators. And so being able to provide that real tractable information on here's our communities if you do X, Y, and Z, and how do they change, and ultimately driving recommendations for management. So absolutely, we are looking at changes over time. When we talk about our metabarcoding data, those are precisely the sorts of data or of results we're generating. Alpha beta diversity, species richness. That's the information we have. In addition to the raw, here are the species that we have in this time and place.
Very nice. Milton asks, I think you alluded to this a little bit, but maybe you could say a little bit more about the nature of it. Is there a physical object, e.g. filter, illusion, that makes sense to database? Like when maybe you could say, you mentioned that you put tags on your samples in your -80. Maybe the question is like, are there 3, 4, 5 classes of things that you put identifiers on that you database? What are those classes of things that you do archive, so to speak?
No, it's really simple. It's two things.
Okay.
We
Yeah.
have two things in the archive. And the first, Milton is dead on. It's a filter.
Okay.
So for most contexts, we have adapted a rinse and filter system. Obviously, with aquatic samples, freshwater, saltwater, we're just filtering that water and we're storing a typically cellulose acetate, although the material doesn't necessarily matter that much, filter in cetyl trimethylammonium bromide. And so when we do flowers, if I want to know what pollinators have attended a flower, we're taking flowers, we're washing them with sterile water, running that water through a filter, and that washes off the DNA extremely effectively. And so we have first that filter. And from that filter, the C tab is going to release the DNA from that filter. It goes into solution, and then we go through an extraction using cetyl trimethylammonium bromide, or sorry, phenyl isoamyl alcohol extraction method that then will release that DNA, and we can use it for downstream applications, either metabarcoding, targeted qPCR. We never use all of that extraction. We always hold some in reserve. So those are the two paired tubes that we have. The filter with the residual C-tab. We never use all the C-tab. And then we have the DNA extract itself. And so
And that,
those are the two physical things that we're maintaining in the -80.
thanks. That's really useful. And that I'm imagining that the tubes could be one to many with the filter in some cases.
Correct.
Yeah.
Yep.
Yeah.
Correct.
Okay.
And so, one thing I've thought about too that, I think one of the exciting things is that as we're thinking about potentially building out mitogenome repositories and sampling from places with conventionally sampling to get actual specimens to accession and to then generate mitogenomes. Pairing that with eDNA sampling, you don't have to extract that DNA, but you could collect at those sites where these specimens are being collected,
Yeah.
and you can harness that DNA and store it in perpetuity in the -80
So
if you ever want to go back in time. And so you can do that. Or I've got a project I'm really excited about. Has nothing to do with eDNA, but we're working on describing a new species of garter snake from Mexico. And ostensibly, we could take environmental samples from the type locality alongside the time where we collected that specimen and include that again in thinking about this extended specimen network. Because once you get it in CTAB off that filter into the -80, it is stable for who knows how long. No one's looked at it yet, but it's ostensibly completely stable over the long run.
Yeah, so you completely anticipated the question that I was leading to. I
Yep.
didn't quite go there, but the idea was that we're thinking about that extra metadata now that somebody goes out into the field and they don't just take a little paper label, right? They want to take some rich data. So when we send out somebody who's bringing back an organism, bringing back a collection object, Sensu TaxonWorks, that's going to be enumerated and preserved, what are the 3 or 4 pieces of data, I guess, and you just listed the physical one, right? Maybe you just go and you swab some leaves, or I'm just saying swab, but you do some basic environmental sampling, and to do that. And so maybe can you say a little bit about, like if somebody wants to go and run some experiments, can they just go by the Mark Davis lab and pick up a little, side bag that has this, and I can just go zip, and then what does that kit look like from a physical Indiana Jones? I'm going out to get treasure, right?
Yeah, that's been one of the more exciting aspects of eDNA as well, is that opportunity for community engagement and community science. And this has been something that's happened for quite some time with eDNA. And if you wanted to do a project like that, if you wanted to sample a whole bunch of places that people are going to, we absolutely make those sorts of kits. And so what they are comprised of is, you can very simply buy hand pumps that there's a collection bottle with a filter flask attached to it, and you can pump water through that. And typically then what we do, it's a little bit different of a process, because as we all know, shipping things like ethanol, shipping things like CTAB can be a little bit problematic. And so in those contexts, what we often do are include gasketed microcentrifuge tubes or whirl packs that have silica packs or silica gel in it.
Right.
We just desiccate it, and then we'll pull that DNA off the filter at a later date in the lab. And so that's what our kits look like. It's simply a hand pump, and depending on, all right, if someone is in a lake management association, for example, and we're taking Secchi dish readings every month, and we want an eDNA sample alongside that, we'll just send enough filter flasks for that May, June, July, August. You've got 4 sets, and throw them in desic at the end of the year. Send us those 4, right? And so very easy to do that. Now, the hand pump is difficult to scale. Individual users that want to take samples from a certain time and place can certainly do that. What we tend to do more frequently with our scientist partners, and we do this a lot, I've been working a lot on DoD installations, is that we'll do some training and we will loan out some of our equipment. And so when we are filtering, we are most often running 6 to 12 samples at a time, because we have these filtration manifolds that have 6 slots. It's attached to a vacuum pump. And so we have these kits that are in Pelican cases, so you can travel with them, and it's got everything you need to do your sampling in a hotel room, for example.
Yeah, neat. So Mark, what about, so that's the physical nature. Is there any compelling, like, sort of digital metadata that somebody could take in the field and add? So I'm imagining, we demonstrated that you can start your collecting event in TaxonWorks by simply dropping a georeferenced image on there, and it pulls off the georeference and it pulls off the timestamp and stuff, and so you're depicting some collecting event with an image, and you're benefiting from the metadata that came from that image in your data capture process. So, even if we had example like a malaise trap, very cool. Thanks
So I
for
said I
sharing that.
wasn't going to do this,
Yeah.
Deb, but here's my colleague Eric Larson. You can see the Pelican case in the back. We are in a hotel room in Rochester, Minnesota, and that's our filtering rig. And so we can deploy those with collaborators. The full rig that we run currently costs about $2,000. The pump, the collection bottle, the filter manifold, and the Pelican case. And so that is something that we can deploy.
Cool.
That if you're a small brown beetle person, this is reminding you of all of the Berlazy funnels that you
Yep.
ran in your hotel rooms, and epic stories about hotels just freaking out because they come into that room and there's all of these, just like literally full of funnels. But
The
this is
number
much
of
more high
times
tech,
the
right?
number of times we've been asked if we're standing up a meth lab in our hotel room is
Yeah,
non-zero.
yeah, I bet. Portable meth lab. Yeah, so back to that digital side of it, right? Like,
Yeah.
is it at all compelling? Like, if you got like a 3D panorama of a site, and then we do some AI detection to classify it, and like, is that going to help, or you've got
Absolutely.
enough little questions already that maybe that's not as useful at the collections level? I'm not sure. Yeah.
No, I think 100%. The more data-rich environment that we can operate in and leveraging remote sensing, the better off we're going to be. And so we were actually just having this conversation in a committee meeting yesterday where, we are looking for very specific types of habitat in stream and along the riparian zone. And so specifically, we're looking for trees that have root masses that are into the bank or large flat rocks.
Uh-huh, uh-huh.
And so the grad student that's kind of leading this project did a great job taking cell phone pictures upstream of the sampling location so we can maybe add some ones and zeros there, but being able to associate with side scan sonar for in-stream habitat, being able to associate with LiDAR to be able to identify tree stand structure in the riparian zone. All of these things are incredibly valuable for us to be able to extend just the nuts and bolts of, all right, we've got temperature, we've got wind
Yeah,
speed,
yeah.
we've got pH, we've got turbidity. Having these data expand what we're able to do, because at the end of the day, most frequently what we're leveraging these data for are to build robust occupancy models. So we
Mhm,
want to be able to understand, number one, what are those covariates that are driving our ability to detect things with eDNA? But more importantly, number two, what are those ecological parameters that are promoting whether or not these species occur in these places?
mhm.
And it's one thing to, dip a probe into the water where we're standing, but having these larger scale GIS data frames that we can
Mhm.
access, and other larger repositories is vastly expanding the types of questions that we're able to
Yeah,
draw
yeah,
and fine tuning those inferences, right?
yeah.
Because the stuff we can take on site with a Kestrel pales in comparison to broader landscape patterns. And as I kind of start off with, our species don't know these geopolitical boundaries.
Mhm,
And so at the point where we're sampling, what's happening upstream in the watershed, it matters a great deal. And, I found this to be a really real eye opener in the Mekong Delta of Vietnam last year,
mhm,
where what's happening in the Chinese end of the Mekong has tremendous downstream impacts on what's going on in the Delta in Vietnam.
mhm,
And so being able to look at a landscape scale and not just at the point of capture is incredibly important.
mhm. And I think that story you could probably tell all around the world, right? That's not just, it might be at a
Exactly.
different scale or a different parameter, but that's not unique to that. Well, there's aspects that are unique, but the pattern is kind of replicable all around the world.
Correct.
Mark, we're coming up on an hour that flew by. I wanted to just ping the chat again to see if there was any last second collections or questions for you. I
I see a
wanted to,
couple asks for kits.
yeah,
Reach
yeah.
out to me. I'm sure that Matt's going to provide my contact information. Reach out and we can chat about kits, how many you need, what do you want, what are you trying to accomplish. Because again, that's the other big, I think, take home point for me. I like to joke that I get phone calls, often crazy phone calls as a herpetologist about a snake in someone's house, but I get a lot of phone calls and emails from our managers, our boots on the ground, who have tried an eDNA study and it has failed.
Uh-huh.
Because you read the literature and it seems so easy, but it is not. And
Yeah.
so I strongly encourage you, if you want to eDNA, contact someone that's got experience in it, because those manuscripts don't tell the whole story. There's a lot of R&D and troubleshooting that goes into it. There are a
Classic.
lot of considerations that may not be made explicit in terms of design and execution that isn't necessarily unpacked in those manuscripts. And so if you want to eDNA, chat with someone that's been doing it, that's got a good track record, because that could make or break your success. And again, this Minnesota Turtle Project that we've been engaged in is the primary example. Minnesota Department of Transportation and Department of Natural Resources really wanted to use these emerging methodologies and technologies to monitor these turtles in Minnesota. And the first time they tried it, it failed. And it failed not by any fault of their own. It failed because, they read this paper and they went to execute, and it turns out that the primer sets had mismatches with the local genetic diversity of those turtles.
Yeah.
And we were able to fix that, and now are looking at going into phase 2 because we've been wildly successful in now documenting the not only detection probability of these turtles, but what are the environmental drivers that are informing occupancy. And so talk to an eDNAologist before you embark on an eDNA study.
I
You won't
think
regret it. Unless it's me, then maybe you will.
I think it's a wonderful little bit to close up on. There's lots more to chew on. I'm excited about thinking about continuing this conversation. Like the people that we invited here are all to Taxonomers Together are all sort of from fields that are kind of guiding where we might head with our biodiversity informatics, or they have aspects of data or their research that we think might be able to link, and sort of we want to be able to magnify what the collections here have in the NIHS with the processes that you run through, with the taxonomy that happens, and like what should we prioritize, what avenues should we go down. So I think this is the start of hopefully a longer term conversation there.
Well, and one thing that we haven't even touched on that I'm also excited about and very germane to the work you are all doing is, I think, really the future and a big part going forward, if and as we're able to build out these backbone genomic repositories,
Yeah.
mitogenome repositories particularly, but also we're moving into nuclear eDNA. But as we build those out, as they're associated with specimens, physical specimens that have been collected that are providing that sequence information, I think there's going to be a lot of opportunity for adapting our bioinformatics pipelines to increase the usage of machine learning and AI.
Mhm.
We're not doing that so much in the eDNA bioinformatics realm right now,
Mhm.
but I think we're going to need to, because as the sequence data continues to accumulate and we're increasingly operating in a big data environment, that is going to necessarily increase efficiencies that we desperately need.
And I think, like you mentioned, even if some of those experiments don't quite work out, the environment is clearly right now ripe for proposing those kind of things and getting funding on those fronts, given the nature of the funding environment that's out there. And so I think there's opportunities on that front too.
Absolutely. Thanks, y'all, for listening to me ramble today.
Thanks, Mark. That was fantastic overview.
Was.
Time flew. Very grateful and to get that overview. And I
Mark,
love hearing about concepts like fade in transport. Like there's clearly some core concepts that it's good to hear. Like, oh yeah, this is just a thing in the eDNA world. And it
Yeah.
was great to get that overview from you. Yeah.
Well, I never planned on being a hydrologist, but increasingly that's a part of my job, for better or for worse.
Yeah.
hello, Matt.
Hi,
Hi,
Doris.
Doris.
Hi, Mark. I'm actually Doris Lagos Kutz. I'm an entomologist. I work at INHS and had the pleasure to work with Mark. As you see him, he's very enthusiastic, he's very cheerful, and people don't be afraid to contact him.
For that
But,
plug, Doris.
and
Yeah.
also about don't get discouraged about when you see a publication and then it doesn't apply to you. You always got, it's very important to reach out to the people that does the work, because like Mark said, there is, 80% is your knowledge, but the on 10% of your multiple, but 10% of the work is a group part, right, Mark? And so that's why I've been writing about, because in my chat, because this is very exciting. It's super exciting work. It's something that we all should collaborate together, because like you say, the backbone of doing this genomic part, but us as taxonomists, to build up this collaborative work is very important, very interesting. I think I'm very thankful, Mark, for being the way that you are, and looking forward to keep working with you.
Well, thank you for your kind words, Doris, and vice versa. Like, I could not be successful without the work that you're all doing, and so please keep doing it, because I'm going to continue to leverage it.
Thanks, Mark. We'll be seeing you around, and hopefully
Yes.
everybody enjoyed that. That was a wonderful second session.
All right. Thanks. Be well, everybody.